So today's talk is part of our scientific masterclass series, where we're trying to create a platform for people to share their research, right? And get some feedback, network maybe with some colleagues. And we're trying to create a platform so things are spread a little bit further than you would usually have with your local networks. The masterclass is kind of centered around the umbrella of biotech research, right? But within that, we have basically a wide variety of topics. It can be membrane protein research, but it can also be protein engineering, for example, or proteomics, or structural biology, or even chemical research. So if you think that you have an interesting story, for example, that you would like to share with a wider audience, right, and then you think this is the right platform for you, then you can just contact us, tell us a little bit why you think your research is connected to Cube, and then we try to allocate you within our masterclass. We have had presentations from people in different stages in their career, right? From PhD students up to very senior researchers. So don't feel afraid that to try to get in touch with us. We're always very excited about hearing what people are doing around biotech research. So let me tell you a little bit about how it's going to work today. So we are going to have our talk, right? After we hear our talk, we're going to have a live Q&A session with the speaker. Right, and after the Q&A session, I'm going to ask you for 2 or 3 minutes of your time. So if you can answer a few questions in a survey, so you can help us a little bit improve the masterclass series more and more. Right, so today's speaker is Dr. David Swainsbury. He is been working for a while now, about a decade, around the topic of how mechanistically photosynthetic protein complexes are helping or are taking over the function of taking solar energy into chemical energy. And he's been doing that by biochemical tools as well as structural biology, right? At the moment, he's a lecturer in the University of East Anglia. And today he's going to be telling us something that is very close to our heart here at Cube Biotech. He's going to be telling us about how he's using SMA polymers to study photosynthetic membrane protein complexes. So without much further ado, I just leave you with our today's speaker.
Postdoctoral and now PI journey to speak today. And what I thought I'd talk about is how my postdoctoral and now PI journey working on photosynthetic membrane proteins has allowed me to demonstrate the real utility of smart copolymers for membrane protein research. And all this has very recently come together in my most recent publication on the cryo-EM structure of the cytochrome BC1 complex. So this is going to kind of be a story of how everything got there and kind of some of the latest data that's come from my research working on these really fascinating systems with this interesting polymer. So first, I should introduce my favorite organism. This is the purple anoxygenic phototrophic bacterium, Rhodobacter sporoides. And these organisms get their name because of the striking red or the purple coloration of the cells, which is due to the strong absorbance of the carotenoids in the bacteria chlorophylls they use to do the light reactions of photosynthesis. Now, taking a closer look at a Rhodobacter sporoides cell, you'll see within these cells are these invaginations of the inner membrane. They're called chromatophores, about 50 nanometers wide. And those house the 4 proteins required to do photosynthesis in these organisms: the peripheral LH2 antenna, the RCLH1 core complex, cytochrome BC1, and ATP synthase. And what happens is light absorbed by these antenna complexes, the energy of that light is transferred to the reaction center, where it drives a series of electron transfers, reducing quinone to quinol, coupled with the uptake of protons from the cell cytoplasm. That quinol then diffuses to the cytochrome BC1 complex, where it's reoxidized, releasing the protons into the chromatophore lumen and transferring electrons to cytochrome C2, which shuttles them back to the reaction center. And that builds a proton gradient that then is used by ATP synthase to generate ATP as an energy source for the cell. Now, for the rest of this talk, we're not going to talk about ATP synthase, but I am going to concentrate on how my work to understand the structure and the function of these proteins and methods to engineer them has also allowed me to understand the best ways in which to utilize SMAR and its real utility for membrane protein research. culminating on studies of this cytochrome BC1 complex here. So of course, working on membrane proteins, we all know that the best place for a membrane protein is within its native membrane, because this gives us the native environment with the correct lipids, the correct protein partners, everything is as it should be. But unfortunately, for many reasons, including the fact that we cannot purify these proteins and light scattering making spectroscopy extremely difficult, proteins in this environment are not amenable to biochemical characterization. So we often turn to detergents to solubilize those proteins, which gives us the ability to study them, but it comes at the price of losing that native lipid environment. And for many proteins, this can be extremely detrimental to the activity and the function of those proteins. Now, to get around these problems, people have tried to have the best of both worlds by reconstituting their proteins into artificial lipid systems, such as liposomes and protein-stabilized nanodiscs. Both of these I use quite a lot in my own research. Of course, this does still require that you solubilize your protein with detergents first. And for proteins that are very delicate and are damaged by detergents, this can really mean that this method is a non-starter. So maybe there needs to be a better way. And when I started working on these systems as a postdoc at the University of Bristol 10 years ago, back in 2012, something quite interesting was happening in the membrane protein literature. There were these SMA copolymers, polymers of styrene and maleic acid, available in varying ratios of styrene and maleic acid groups and various polymer lengths. And it turns out that if you expose lipid membranes to these polymers, they spontaneously create lipid nanodiscs. And this even works on biological membranes, allowing you to solubilize membrane proteins with a belt of their native lipids all stabilized by this polymer directly from their native membranes, meaning they never see detergent. And really, to be honest, I thought at the time this all sounded far too good to be true, because it gives you everything: the native environment, the ability to purify the protein, and it allows it to be amenable to most biochemical techniques. So at the time, I really wanted to know, does SMA live up to these very high expectations? And doing that, I worked with the purple bacterial reaction center, which we can generate in a native membrane through genomic removal of the LH2 and the LH1 antennae. And this is a perfect model membrane protein because it doesn't interact with any other proteins within the membrane, meaning that it's very easy to work with, very easy to solubilize. And it has a few other distinct advantages. The bacteria chlorophylls that it uses to absorb sunlight, their absorbance is out in the infrared region of the spectrum and is not particularly affected by light scattering, meaning we can still do biochemistry in the native membranes. And the protein is stable in a wide range of detergents, allowing us to see the effects of different detergents on the biochemistry of this protein. So maybe these advantages would allow us to look at SMA and see what its effect on the membrane protein properties were as well. And so I went over in 2012 to visit Stefan Schindler, who was working in Antoinette Killian's group at the University of Utrecht. And through a lot of trial and error, we eventually managed to purify these bacterial reaction centers using SMA, settling on polymers with a 2-to-1 ratio and a relatively low molecular weight of 10 kilodaltons in the end. But the big question is, do these SMA preparations of the reaction centers really live up to those expectations? And so doing a few experiments, it turns out that they did. We could see that the lipids co-purifying in SMA, as you can see on these TLC plates, resemble those in the native membrane, and about 150 lipids were purifying with each reaction center. Whereas with the detergents DDM and LDAO, those lipids were almost completely removed. By negative stain TEM, we saw the nanodiscs were about 12 nanometers in size, and labeling with these nanogold labels, we could see that the reaction centers were integral to those nanodiscs. But what about the biochemistry? So I'm not going to go into the details of these experiments. I just want to concentrate on the similarities and differences between the traces. But hopefully you can see here when measuring the redox midpoint potential of the bacteria chlorophyll special pair, the red trace of the SMA purified protein resembles almost exactly that of the purple trace of the protein in its native membranes, whereas there is a significant shift for the protein purified in detergent. And the same is true for these charge recombination assays, essentially a way we can look at photochemistry within the reaction centers, whereby the protein in its native membrane and purified with SMA looks rather similar, whereas there is a significant shift to much faster recombination times purifying in detergent. So these experiments show that the biochemical properties that are altered with detergent solubilization of these reaction centers are very well retained in smart nanodiscs. Now, at this point in my career, I had already become convinced that using SMAR to purify proteins where those proteins are amenable was really the way to go. And for the most recent study I'm going to get onto in a few slides' time on the cytochrome BC1 complex, nobody was convincing me that detergents would be the way forward. But before getting to that, there was something else that I really wanted to understand about these SMAR polymers. And that's what happens when you apply them to a more complex membrane system. So when I moved to the University of Sheffield for my second postdoc, I started working on more native complexes where all of the proteins were present. And these chromatophores are rather interesting, as you can see from this computational model. These LH2 and RCLH1 complexes form protein-rich domains. They form weak protein-protein contacts that hold these proteins in close proximity to aid energy transfer through this system. Whereas the cytochrome BC1 complexes do not interact with any other complexes and are surrounded by a small patch of open lipid membrane. So they are residing in lipid-rich domains of the chromatophore. So I got wondering, what happens if you use SMAR to look at the proteins within a complex membrane system like this? And it turns out what happens is SMAR selectively solubilizes the cytochrome BC1 complexes in those lipid-rich regions, whilst the protein-rich RCLH1 and LH2 complexes are very poorly solubilized. And actually, it turns out that these proteins that we're recovering in these assays are in large membrane patches that survive ultracentrifugation. They're not actually truly solubilized into individual complexes. So it appears then that SMAR is very delicate on weak protein-protein interactions, keeping them intact. And it also turns out that it can probe the local lipid environment for protein, telling you whether it is in a lipid-rich or a protein-rich domain. And this is in stark contrast to beta-DDM, which indiscriminately solubilizes everything. Now, as I was doing all this work, I had a growing interest in this cytochrome BC1 complex, partly because the cytochrome BC1 complex, as I'll come on to in a moment, is a really interesting and unique complex amongst this family. Now, this family of proteins is found in many species of bacteria, and it's a close relative of mitochondrial complex 3, because the alpha proteobacteria, to which Rhodobacter sporoides belongs, are the progenitors of the mitochondria. And these proteins are more distantly related to the cytochrome B6F complex of plants, algae, and cyanobacteria. So they're essential to many different electron transport chains. And from the perspective of improving photosynthesis, they are the rate-limiting step in photosynthetic electron transport. So if we can improve the activity of the cytochrome BC1 complex, you can improve the photosynthetic capacity of these organisms. And this has been shown through several computer simulations. And a recent study in plants where B6F was overproduced led to an increase in biomass yield from those plants under real-world field conditions. So all cytochrome BC1 complexes share a common structure and a common mechanism. And that common structure is these three core catalytic subunits: the membrane intrinsic cytochrome B, which binds two B-type hemes; cytochrome C1, which has a soluble domain in the lumen or the periplasm, binding a single C-type heme, and that is anchored by this single transmembrane helix running behind the complexes we're viewing it here. And this risky iron-sulfur protein with a soluble domain in the lumen binding an iron-sulfur cluster and a single transmembrane helix. And you can see that that's reaching away from the rest of the complex. And the reason it does that is because all cytochrome BC1s are dimeric, and this domain swapping with the head of the risky domain in one subunit and its transmembrane helix reaching across to the other one, interacting with the C subunit and the B subunit within the membrane, is one of the main interactions that brings the two halves of this complex together. Now, for the simplest cytochrome BC1 complexes, this is all there is, these three core catalytic subunits. But some cytochrome BC1s have become much more complex over the course of evolution, evolving supernumerary subunits. For example, your mitochondrial complex 3 has 8 supernumerary subunits, giving it 11 subunits in total. And this is where the Rhodobacter sporoides complex is unusual, in that it has a supernumerary subunit called subunit 4 that is found in just a few of Rhodobacter sporoides' closest relatives. Other species do not have anything in their genome with any similarity to subunit 4. But this is where things get really interesting, because that fourth subunit is missing from every single PDB structure that was out before conducting my most recent work. And the reason for that is that subunit 4 is labile in detergent, and it is lost during the process of purification and crystallization, meaning that the subunits that crystallize are just these 3 core catalytic subunits. And this got me wondering, was SMA the way to get the 4 subunit complex, its structure, for the very first time? So going back and looking at the properties of those cytochrome BC1 complexes I was able to extract from chromatophores, I think you'll agree with me that the answer is definitely a yes. Subunit 4 is retained in stoichiometric amounts with the 3 core catalytic subunits. About 30 lipids are co-purifying with each cytochrome BC1 monomer, that's 60 per dimer, along with 1 of the native quinone substrates, with a similar profile to that of the native membrane. And by UV vis spectroscopy, we can see that the cofactors are retained in the expected 2:1 heme B to C ratio. By native page, we can see the complex is dimeric, and this is also confirmed by negative stain electron microscopy, showing that the nanodiscs have the consistent size and shape for a dimer. And I think this really neatly shows how the protein is templating the size and shape of a smart nanodisc, not the polymer, which I think is a really neat feature of this copolymer system. Now, unfortunately, at this point, I had excellent preps of the protein, but the yields were extremely low. And with cryo-EM time limited, I had to concentrate on other projects for some time. And then it was time to finally get going on this project when, unfortunately, the COVID pandemic hit and everything ground to a complete halt. But fortunately, after the pandemic and the lab was opening up again, I was joined by a very talented master student, Sabina Musial, whose job it was to essentially start again with this project and go back to my preparation protocol for cytochrome BC1, tear it up, and re-optimize every last step. And that's exactly what she did. She re-optimized growth of the cells. She re-optimized the nickel affinity step. Importantly, we found that we had to reduce the imidazole concentration during washing to just 10 millimolar to prevent early elution of the complexes. She then developed this sucrose density gradient step to enrich the cytochrome BC1, removing the contaminating proteins. And then finally, after polishing by size exclusion, she could show me an Eppendorf tube with deep red cytochrome BC1, more than enough to do the biochemistry and structural biology that I'd really wanted to do for a long time. So the first thing Sabina did was stop flow assays, showing that the protein in SMA is indeed active. This is looking at the reduction of cytochrome C2 in the presence of excess quinol. And this rate of activity is similar to the protein in native membranes and freshly prepared with detergent before it had the chance to fall apart. Now, she was also able to purify a protein from a strain where the lab technician, Liz Martin, had deleted subunit 4. And this clearly shows what subunit 4 is doing. It raises the activity of the complex roughly threefold. This has been seen by our experiments and by others working on subunit 4 beforehand. And what it's showing is that the subunit 4 effectively raises the photosynthetic capacity of the cells, because you'll remember that BC1 is the rate-limiting step in photosynthesis, with the relatively inexpensive addition of a small polypeptide subunit. Whereas the only other way to do this would be to have 3 times more cytochrome BC1, which would be metabolically extremely expensive. But these experiments do not tell us how subunit 4 is working. And to do that, we needed a high-resolution cryo-EM structure. So we prepared cryo-EM grids, and hopefully you can see the individual cytochrome BC1 particles within this thin layer of vitreous ice inside this quantifoil hole on the grid at 73,000 times magnification. And these grids were sent to our collaborators at EBIC, the National EM facility in the UK, based at Diamond Light Source, where Fred Hawkins collected nearly 16,000 images of this complex for us. From that, we could extract nearly 4 million particles to take through a standard cryo-EM processing pipeline, getting a map in the end with a resolution of 2.9 angstroms that looks like this. And you can clearly see the cytochrome BC1 complex embedded within this diffuse density corresponding to the lipid nanodisc, which is less well ordered. Now, using that to place the membrane and then focusing only on the well-resolved regions of the map, you can clearly see the features of this protein. And it was trivial to fit the existing 3 subunit crystal structure into this map, leaving these regions of gray density that were unassigned, one of which looks very suspiciously like an alpha helix. And that alpha helical region corresponds exactly to the expected sequence of the predicted transmembrane helix of subunit 4. And now these remaining areas of unassigned density correspond to bound lipids and that quinone molecule I told you co-purified with these complexes earlier on. In cartoon representation, we can see what subunit 4 is doing. You can see it's lying across the transmembrane helices of this risky domain and of the cytochrome C1 subunit. And you'll remember I told you that this interaction of the risky domain reaching across the dimer is one of the main things that brings the two halves of the complex together. So I like to think of subunit 4 acting like a protein staple, to staple these two parts of the protein together, increasing the stability of the dimer interface and therefore the protein as a whole. And this translates to the increased activity I showed you in those stop flow assays earlier on. And we also think there may be some conserved residues at the bottom of this subunit 4 as we're viewing it that help to guide quinones into and out of this central cavity. Now, with the last couple of minutes, I want to show you something else quite interesting that came from analysing these cryo-EM structures. And that is we were able to resolve movement in the risky domains of the protein. Now, this was done by a method called focused refinement, in which you take the particles and mask away signal corresponding to anything outside of a region of interest. In this case, the region of interest being the risky domains. And in the consensus class, that is the model built, the map built from all of the particles, you can see both of them are pointing downwards in this view. But by taking those masked particles and sending them back through classification, we were able to get one class of particles corresponding to about a third of the entire particle stack, where one of the risky domains has adopted a different conformation to that of the consensus class, which hopefully you can see in this overlay. Analysing the models that were built, you can see that this is due to about a 52-degree rotation of that risky domain, taking it away from this B subunit down here and bringing it close to the C subunit up here. And this conformational change has been observed before through inhibitor studies and crystallography and through some more recent cryo-EM studies, and is known to be a central part of the Q cycle by which this protein operates. But what's new and interesting in this study is asking why only one of those two subunits is moving. And it turns out the reason is that one of these QO sites to which the ubiquinone binds, there is a ubiquinone molecule, whereas on the other side of the complex, there is no ubiquinone bound. And it turns out on the side that is bound, that's the stationary side, and the side with the apo-QO site, that is the mobile side of the complex. But why is the quinone bound state stationary? Well, looking closely, it turns out that this quinone is forming a hydrogen bond to this histidine residue as part of the risky subunit. And that hydrogen bond effectively locks the protein, the risky subunit, in place, preventing it undergoing its conformational changes. Whereas on the other side of the complex, there is no quinone bound, so there is no hydrogen bond, and that risky subunit is free to explore its range of conformations. And this is the first time that that conformational change had been directly observed to be correlated with the binding of a native substrate, because before now it had only been inferred through inhibitor studies. So with that, I'd like to thank you all very much for your attention. I'll leave you with my slide of acknowledgements. Many of the people on here I've already mentioned, and I look forward to answering your questions.
So thank you very much for the talk. That was quite interesting to hear. We always love to see things done with polymers. Just a few things before we start with the Q&A. So there on top, there is a link to the paper of the research that David was presenting right now. So if you click on it, then you can go directly to the paper. If you have questions, just put them in the chat. So there's a specific icon there that is like a question mark. So you can label your questions for the Q&A, so we know it's a question for David, and then we can pass it along. And perhaps I can start with the first question. So you in the beginning you said that you were trying different SMAs in the beginning to see which ones were working the best, and then you chose in the end, I think, Xeron or one of those. What led you to that decision? So was it purification efficiency what was lacking, or was the protein not being active enough once you purify them in other polymers?
So one of the first things we did, using the fact that we can do spectroscopy on the proteins in the membranes, was to do a fairly simple assay looking at the solubilization efficiency. So it was a case of taking the membranes, treating them with SMAR, ultra-centrifuging everything and then checking the spectrum again afterwards to work out how much of the protein from the initial membranes had made it into the solubilized smelt. And it turns out that those 2-to-1 low molecular weight polymers were the most efficient of the set. Although out of the set that worked, it was all actually rather similar for the 3-to-1 and the slightly higher molecular weights. But there's also the handling of those. The lower molecular weight polymers tend to be soluble at slightly higher concentrations for making stock solutions. So it was a mixture of the best for solubilizing and a little bit of practicality on top.
Right. But the ones that you had solubilized in other less efficient polymers were still kind of properly folded and working as expected, or?
They seem to be, yeah. The signature of it's a spectrum, so those spectra in the infrared region is extremely sensitive to the protein's environment. If it unfolds, we see it immediately. So we didn't go any further than looking at the spectra, but the spectra that we got all looked identical, which would infer that the biochemical properties are likely to be very similar.
Right. Just the last question before we go to the Q&A. I'm very curious about this density gradient, that step that you had in your purification. I see it in people doing membrane research as well. Was there a specific reason for you to do that? Or
so we've used it for a really long time in detergent solubilization to separate out all of the complexes. So the RC-LH1 core complex and LH2 and BC1 will all separate out once you've solubilized them essentially based on the mass of the proteins. And one of the problems we were having after the nickel affinity step was a small amount of contamination. It's largely from these large membrane patches of RC-LH1 and LH2. Right. And so the same method based on the mass, those large membrane fragments are so heavy, they go straight through the gradient and pellet. Whereas the first-chrome BC1 complex was separating away from those and a few other contaminants quite neatly by its mass. And it was a method to get around the fact you can't do ion exchange with these polymers because of irreversible binding to the resin. So the sucrose gradient was essentially a replacement for what I would normally use ion exchange to do.
Right. Cool. Okay, then Carmen Castillo asks, what makes Rhodobacter a model organism? Why were you interested in studying photosynthesis with it?
So I could list 100 reasons for this. One of the main reasons is it's genetically tractable. So doing genetic modification, making mutants, deleting proteins, putting new proteins in is all relatively straightforward in this organism. We've been able to do that for a very long time. Its photosynthetic machinery is related to that of more complex oxygenic phototrophs, but is much more simple than the more complex phototrophs. So it gives us a really good window into how photosynthesis more complex organisms work. And it's the ideal test bed to develop new methods and new technologies and really see how they could be applied later on. And one of the big things it does is it can make its photosynthetic complexes, but it doesn't actually need to grow photosynthetically because it can grow by respiration. It can do things like fermentation. So it lets us make really drastic changes to the photosynthetic machinery that in most phototrophs would be lethal, and then see biochemically why those changes to the photosynthetic machinery are having the effect they're having. So it really opens up the possibility of experiments we can do.
Cool. Right. So Ellis Thornhall asked, but what does it mean for every different membrane protein? I need another SMA. I think this relates to the slide that you have where SMA was sensitive to the membrane and to the specific environment around the protein.
So the polymers that I've tried all tend to behave relatively similarly on this system. I think what's actually going on is the reason it's not separate, it's being specific in its solubilization, is that the protein-protein contacts between RC-LH1 and LH2, they're very weak, and that's actually the native state of the protein. And I think what's happening with DDM detergent, and well, LDO actually destroys the proteins completely, but with DDM, the detergent molecules are able to substitute not only the protein-lipid interactions, but those hydrophobic protein-protein interactions as well, which is why it breaks them into monomers. Whereas it seems that SMAR is just not interacting with the protein at all, and it's leaving those native protein-protein interactions behind. So I've not done an awful lot with the membrane fragments, but we did some experiments like atomic force microscopy, and they look remarkably like the native membrane. So it seems that it might actually be, rather than calling it a big insolubilized area of membrane, they might be giant SMAR nanodiscs that are containing arrays of closely linked membrane proteins.
Right. So Brigitte Lohnholzer asked the cryo-EM sample preparation. Were those still SMALPs that you were using?
Yeah, so once the protein is initially purified in SMAR, something I forgot to mention earlier on in the talk was that once you've done the initial solubilization, the SMAR nanodiscs are completely stable without adding anything else to the buffer. So you start handling these nanodiscs like they're a soluble protein. There's no extra SMAR, no detergent or anything else. So yeah, once they were purified in SMAR, they remain in those SMAR nanodiscs, and that's the same sample that went through activity assays and then onto the EM grid.
Cool. So Lynn Brown asks, any theories as to why the BC1 dimer only binds one ubiquinone peptide? Is it a homodimer, so it should fit into the ApoQ0 spot just as fine?
Yeah, so it is a homodimer. So there was the same three subunits are mirrored with C2 symmetry. Why it was only on one side is that's a bit interesting. So one of the reasons it's a dimer is thought to be that central cavity helps the quinones go through. I didn't go into detail of the Q cycle, but it actually binds a quinone. And then once that quinone has been oxidized, that quinol has been oxidized back to a quinone, it binds to a secondary site within the cytochrome BC1. And the cavity formed by dimerizing them can trap them and help the quinone shuttle between the Q sites within the protein. Why we found it only on one side, I think it's probably luck for the most part. We often get quite fortunate with substrate binding. And something that we did was deliberately solve the structure without imposing symmetry on it, because symmetry is optional in cryo-EM in the hope we'd see some asymmetrical features. There was density in the other quinone binding site, the QI site, which is probably a quinone bound there as well, but it wasn't well enough refined or well enough defined to be able to confidently fit anything into it. So it was left open. But yeah, it's partly luck to do with the quinone occupancy and some of the inherent asymmetry, where it seems that one side of these molecules has some sense of what's going on in the other side, may mean that one of the quinones was filled while the other one was empty.
Right. Torsten Uriel asked, your BC1 complex were his tagged due to referring to a nickel affinity step. I heard that his tags are not the best option for membrane protein purification. Did you try something different as well?
I didn't try anything else. So we'd already made a his tag strain of the cytochrome BC1 for several other experiments, including actually labeling with those nanogold labels that I showed you earlier on.
Right.
And the other reason is that with SMAR, the purification tends to be the rate limiting step. A lot of people attempt to purify these proteins and give up because they find that it's not binding to the column. So I've had some limited success with strep tags with other proteins. I've tried flag tags. Some people have got it to work with SMAR. I haven't, but I've not tried an awful lot with flag tags. But really, the reason for sticking with his tags is I've found that I can get that reasonably consistently to work. Although there are a couple of caveats with it, which people might find interesting. One, the binding to the nickel affinity resin is extremely slow. So batch binding overnight or recycling over a column to get the protein to bind has almost always been needed for me. And as I mentioned earlier on in one of the slides, keeping a very low imidazole concentration during washing is needed to get the his tags to work as well.
Right. So it starts also eluting very early if you do a gradient, right?
Yeah, I think the polymer interferes with binding. It's like having a high concentration of imidazole in the binding buffer already.
Right. This is something that we've seen here in the lab as well, that you have to, if you want to work with his tags, then you have to kind of work around it a little bit when you have polymers in the sample. So perhaps we have time for one last question. And so Brigitte Lohnholzer again she's asking about why is absorbance curve flattening over time? I think it's related to this slide where you were measuring with and without subunit 4 over time. If you can explain a little bit more that experiment.
So what we're seeing there is the experiments are done. So there's no proton gradient because the nanodiscs are free in solution. So there's no barrier, no membrane for them to build or no continuous membrane, should I say, for them to build a gradient across. So what's happening is we take a low concentration of cytochrome BC1 complex and add an excess of quinol, which is one of the substrates and oxidize cytochrome C2, which is the other substrate, and then watch the absorbance change at 550 nanometers for the cytochrome C2 heme over time. And there's a lag phase at the start, some people often notice. That's to do with a quirk of the Q cycle, where you need both quinone and quinol present for things to work at full activity. Then you go into the steady state phase, and as the curve flattens off, that's just it running out of substrate. So the substrate concentrations are effectively dropping, the reaction slows down, and eventually a substrate completely runs out and everything plateaus.
Right. Okay, super. So if any of you have more questions for David, just leave them in the chat or drop us an email, and we are going to make sure that all the questions are answered in the end via email. Thank you very much again for joining us today, David. That was super interesting again. And then
thank you for inviting me.